everybody can see on their display. So I was trained as an evolutionary biochemist and was really pulled into the food allergy research. Personally, I'm very interested in this topic, but this organization really provided me and my lab a great channel to collaborate with researchers from many different disciplines, immunologists, biologists, people in the medical fields, physicians. So I really enjoy this exercise. So today I'm going to share with you some of my thoughts coming from a chemist thinking about food allergy as a disease, really at the interface between human and plant interactions. So one thing when I think about food allergy is the question that what would be the ideal food for human beings? So we as the species of Homo sapiens, the modern human, arose on the planet around 300,000 years ago. So this is actually pretty recent. And we were a species of hunter-gatherers. So for the majority duration of our species, this is the lifestyle we live on. So the picture you're seeing are the Hadza people who still survive and live in Tanzania. They're almost the last living pure hunter-gatherers. So they go on and hunt for animals and have some success, but not every single day. But most of the time, will forage in plants they can gather. So these Congolope berries are the ones they pick along the way, chewing and then spitting out the seeds. So the plants will grow the next year. So the same thing still also happen in South America. So these are another different native Indian tribes living in the Amazonian jungles. And there are about 100 villages that kind of are still there along the river. And the native people will collect plantains from the trees and beans from the Inca trees. So these are the most of the food they eat today. And we can reflect what we are eating today in our modern diet. I would say it's quite different. Something quite dramatic happened around 10,000 years. So this is when the agricultural technology was invented. So it was really the people living around their settlements, finding species of wild grass, and their seeds can be eaten. And by growing these grasses, people realized that you can store the seeds and eat in a later time. So then agriculture emerged independently in different continents, in Africa, in Asia, and also later in other places around the world. So that invention has permanently changed the way we eat food and also changed the way we live our life. So we become really settled in one place because now we have surplus in food. And the main calorie intake now instead of coming from much more diverse plants, coming from only a few species of grass. So wheat, rice, millet, and some other species. And here are some prehistorical records for that. So this is these kind of clay plates are the earliest record for food supplies, because there's the surplus, and then you have to record how many food have each family produced and being recorded in these things. So really to think about, this is kind of how human civilization has evolved, because it's emergence of agriculture in a way which was really captured by Yuval Harari in his book, A Brief History of Humankind, talking about domestication of wheat. He really thinks about it's the opposite, that the wheat probably has domesticated us to grow them, so that we work very, very hard to just get that surplus. And actually in the last couple hundred years, the industrialization is the extension of that, because we can work harder, generating more wheat, and thinking about modern human beings, we can work harder and create more wealth and create more products, and eventually leading to the modern life we live in. So in the long history of human beings, the lifestyle we live in particular the food we eat, only came about in the last 100 years. So this picture really captures the more Americanized the food that's kind of being eaten today. Much of that is processed. And really the point here is our body, our genetics has never adapted to this. So this is too soon. Our body really adapted to a Paleolithic diet. So our genetics really adapted to mostly plants and maybe occasionally meat and a much more diverse varieties than what we eat on a daily basis. So then kind of coming back to a little bit into plants. So I study plant metabolism, and I really think about how plants evolve over time. And the time scale for that is a lot longer than humans. The first land plants emerged 470 million years ago. And that was really a transition from aquatic plants to really adapt to terrestrial environment. And later on, different major lineages of plants emerged. So when we talk about major food plants, and that was really coming about around 200 million years ago, the foods that cause food allergy, wheat or peanuts, are late inventions in the plant kingdom, but still they're hundreds of million years before human even evolved. So it's kind of a very different mindset. You can think about this and think about a plant life. What does plant want? It wants to live there doing photosynthesis, and they really want to defend themselves against harms, herbivores or pests. So over the time, they become very good at doing this. And this is really at the essence of what my lab does, is to find those evolutionary traits that make plants to live better. So this is Australia stinging tree called Jing-Ke. This is a plant that we actually study. We found that they make this very painful molecules, and these molecules are made in those trichomes, these needle-like structures on the surface of the leaf. If you get stung by that, that triggers extremely pain, a quite dramatic immune response to it. So essentially, plants is master of defending themselves. So I think that's quite relevant when now we think about food allergy. So the way I think about food allergy, I think much of that condition is thought as a disease, but I think it is really, it's a manifestation of a highly evolved mechanism that for our body to defend against things that are potentially harmful for us. Now we think about what are the source of allergen proteins. Most of that coming from plant seeds, peanuts, tree nuts. What's special about those? And these are really the most valuable genetic materials for plants as they pass their offsprings from one generation to the other. So maybe what we think is there are certain proteins that are made in the seeds. They have to be nutritious so they can support the germination of the seeds when they need to. But they also serve as critical protective mechanisms for the plants to survive. So that's why they have manipulated the human immune system so that for a lot of us, eating those will trigger an immune response. And with a lot of studies now, we know that response, the IgE mediated pathway, is overlapping with the parasite, our body responding to parasite. So really that food allergy is not just thinking about the human perspective, it's really at the interface between plants and human. So plants have really evolved to trigger things like this so they can protect their valuable seeds not being consumed at large quantities by animals. So the other aspect about food chemistry that Chris has mentioned is to do with the complexity of chemistry in plant food that we really, even until today, had a relatively rudimentary understanding about. So this is the typical nutrition facts label you would see on foods. And for broccoli, it'll simply tell you, what's the calorie, what's the fat content, what's the polysaccharides content, and maybe some vitamins. But it's really a underrepresentation of the true chemical diversity that's present in plant food. So nowadays, we can use high throughput liquid chromatography mass spec technique to measure and quantify the chemicals that are present in plant tissue. So this is a simple analysis of broccoli extract. And the compounds present can be separated based on their molecular weight and their hydrophobicity and be detected by the mass spec. And every dot is a metabolite. And there are literally hundreds of thousands of metabolites that are present, and we know at most 5% of them. So some abundant ones like glucoraphanin and some other glucosinolates are the health promoting molecules in broccoli, but we essentially do not know what the other dots are. So these are really kind of the dark matter of food chemistry. As we move along, hopefully we can gain more insight into this. So at the interface between plant and human interaction is our body's ability to perceive very precise physical information around our world. So we have a lot of sensory mechanisms to see light, to hear, to taste things, to smell things, and also to have feeling of touch, itch, and pain. So many things are well studied, and the topic that my lab is particularly interested in is the chemical modulations of these sensory mechanisms. So even though some mechanisms evolve to perceive just a physical cue, but usually these receptor proteins can also be modulated by small molecules. Many of these are present in plants naturally. So that interface really provides tremendous opportunities to study potential mechanisms, molecular mechanisms, and also maybe come up with cures that can treat disease. So here is a very well-known example for that. So TRPV1 is an ion channel. So it's a membrane localized protein. Its normal function is to perceive physical damage or heat. So that's the main mechanism. When you touch a hot plate, you will retract your hand. So this can be really manipulated by plants. There are two plants that produce chemical signals. One is chili pepper producing capsaicin, and also resin spurge produce racinoferritoxin, a molecule that's 1,000-fold more spicy than capsaicin. So these molecules bind to specific sites on TRPV1 channel, then trigger massive pain. Spiders evolved independent mechanisms to do things similar. So if the pain receptor can be manipulated by plant small molecules, how about the receptors that are related to allergy? So this is kind of more a personal story. Last summer, I was handling some arborvitae trees that got scratched in the skin. In the same day, I developed mild rash, but then in the following 2 months, I was suffering this skin allergic reactions that took almost 2 months to resolve. And eating certain foods makes things worse. Through interaction with FOSSI physicians, I know I got chronic idiopathic urticaria. So it's not food allergy, I learned, but it is a peculiar reaction of our skin to some phytochemicals that's in the trees. So the same things could occur also in the gut. So this is really get into the research that's sponsored by FOSSI that's currently ongoing in my lab. So we are hypothesizing that there are specific receptors in our gut that are in sensing, constantly monitoring the food qualities, the quality of foods that we eat. And then the reactions we will respond to these foods, and specifically the molecules present in the compounds, will then dictate whether the food should be discharged. So really, it's the very well evolved system for our gut to respond to noxious substance. In the context of food allergy, so we know there are present of antigenic proteins, like the seed storage proteins in peanuts. And there are many other phytochemicals that may be co-present in these foods. So over time, if one type of food, that protein, triggers allergic reactions, over time, our immune system and neurological system may develop sensitization mechanisms so that at a certain time we'll also sense the presence of these bitterants. So these phytochemicals that are coexisting with antigens will exacerbate the later on food allergy episodes. So this is at the heart of the research we want to conduct. We're still at the early days. We know bitter compounds by themselves do not trigger allergic reactions because we drink coffee and rarely people are allergic to coffee. So we're studying the genetics of that. We're also creating the bitter taste receptor assay so we can screen a large quantity of molecules and finding the ones that may be interesting for us. We're also looking into the modern human variations and early human data and finding single nucleotide polymorphisms that may define different response of people to different bitterants. Working with other FASI scientists, we are putting together a list of plants that people may develop food allergy to, and then really assign the family of phytochemicals to these plants, to seeds, so that we can start to recognize the pattern of molecules that trigger food allergy. The last couple minutes, I also wanted to say that plant food not only are the source of allergy, they could also be a remarkable source for future therapeutics. So we know that herbal remedies have been utilized around the world to treat all kinds of disease. And in the last 100 years, single molecules that had been identified, isolated from these medicinal plants that have become very important medicine. So through our work, we really want to find future therapeutics for treating food allergy. So over the past few years, I had the honor and pleasure to work with Dr. Xiuming Li at New York Medical College. And she has really spearheaded in her career to use traditional Chinese medicine for treating food allergy. So there was a very ancient recipe that was developed in the 1st century by Zhongjing Zhang in this this collection of recipes for treat cold damage. In one of them, and Xiuming Li's work has identified, this is a recipe initially developed for treating diarrhea and also malaria, things that are really the allergic reactions that's overlapping with food allergy. And her work has resulted in the development of FAVE2. So it's a recipe of 9 different components, mostly from plants, and she has utilized this in her practice. And I've seen many patients have been treated by this. So we've been looking into the recipe and finding specific plant molecules and also fungal molecules that could become interesting candidates for treating food allergy. So some of these compounds are listed here. For example, berberine, alkaloid that's coming from 3 of the 9 ingredients in this recipe. We're also looking into specialized steroidal compounds from Ganoderma mushrooms, which not only potentially have a role in food allergy, but also in asthma treatment. So we're taking synthetic biology approaches so that we don't have to eventually rely on the plants or the fungal. We're trying to figure out the specific genes required for making these molecules, so we can not only reproduce these molecules in a heterologous system, but we can also derive analogs of these molecules, and all of these can then be fed into a drug discovery effort. Hopefully, we can deliver modern versions of these therapeutics in the future. So before I close, I want to really thank people, researchers in my laboratory who did the work, especially Kate Higgins for focusing on the bitter taste receptors, and Tomo Kenda working on looking into steroidal synthesis in Ganoderma mushrooms. Really a big shout out to the funding support from FOSSI and from the Draco Family Foundation. And also I really cherish our collaborations with Ruslan's lab, Seth's lab, and Xiuming Li's lab. And without these collaborations, these wonderful science would not happen. So I'll stop here and happy to answer any questions you may have. Thank you.